Mic check 1, 2. Alright, we're good on the audio. So hello everyone, my name is David Rock. I'm here at the University of Maryland in Baltimore, Maryland, and welcome back to Cytometry in R. And today we're going to be talking about how do we carry out unmixing in R. So this is now the 14th week of the Cytometry in R course as it was originally envisioned. In the course of this walkthrough, we're going to gather the various bits of coding knowledge, building blocks that we have been tinkering with since week 9, and attempt to unmix our raw full-stain FCS files using the spectral signature matrices that we have derived from our mixing controls, both single color and unstained. We will also make some necessary formatting changes to the unmixed FCS files so that we avoid visualizing our data as teeny tiny distant dots when we open them later on using the commercial software. More importantly, the goal for this lesson is we hope to crack open what to many in the community seems as a black box. By understanding the underlying mechanics operating behind the curtain, we can gain a greater appreciation of the elements involved in the unmixing process, which will hopefully assist us with contextualizing/troubleshooting any unmixing errors that we may encounter in the future. The goal of this primary course walkthrough is focused primarily on providing a broad, detailed overlook of the unmixing process. Additional details on unmixed FCS file reformatting will be provided in a separate bonus walkthrough for those of you who are interested. In the upcoming week's primary walkthrough, we'll actually be going through how to systematically evaluate your unmixed FCS files to be able to find mixing errors, and then make the decisions as far as what signature variants do you want to use in your unmixing reattempts. Additionally, there are two separate community walkthroughs for both AutoSpectral and TrueOS currently available on our website, providing alternative unmixing approaches that are already present. These were initially designed with just a 4-color panel in mind. My goal for this weekend is to revise them to actually feature our 29-color example we're using today, so that way they are directly comparable to this lesson. And with that, let's go ahead and get started. So for unmixing, we're going to be working with raw full-stained FCS files. These can be retrieved from the SDY3080 import repository dataset, the same place we've been gathering for our single color and our unstained unmixing controls over the last several weeks. Since these original files are rather massive in size, I ended up downsampling them for the course to allow us to share them back and forth with GitHub without hitting the file size limit. These downsampled FCS files are what you can find in this week's data folder, so at this point you just need to go ahead and sync your fork of the class, pull it locally, and then you'd have access to these files. In addition to the full stain samples, we'll be needing the signature matrices that we have derived from our mixing controls, both single color and unstained, from the previous week's walkthroughs. An important thing to note is we have not yet fully validated these signatures. If you think of the bead controls, we haven't actually seen if they're bright enough compared to the cell controls. Similar story, some of those cell signatures we derived have a lot of autofluorescence/tandem degradation contributions, so we don't actually know if they're going to work well when it comes to our unmixing. However, the goal of this particular walkthrough is mainly just to figure out how to do the unmixing process, so the actual evaluation is something that will be happening in the next session. So for now, these little intermediate signature matrices should be more than good enough for what we need to do today. And as always, you're welcome to follow along using your own FCS files. The dataset we're using today was acquired on a site like Aurora. The main thing if you're using a platform from a different manufacturer would be the choice of keywords. So in this case, later on when we do the file renaming, we're using both group name and tube name tube name is not actually found in all different manufacturer keyword lists, so you may need to vary that a little bit here and there. And then even on a site like Aurora, most of the instruments defaults are just going to be acquiring -a, meaning area, but you have the option to also be including -height, -width as well, depending on what you selected in your instrument control setting. So this may not even be universal for all Auroras, so these are a couple things to just be aware of before you go and try your own data set. Consequently, if you do try your own data set, there may be small changes that are needed to be carried out to the existing code to be able to account for these differences. So if you get stuck, or you want to share your solutions for the different manufacturer instruments so that others may benefit and you don't have to reinvent the wheel, please go ahead and post those on our discussion page, and we can help you navigate for our own respective instruments. And with that, let's go ahead and begin the journey into the black hole that is spectral unmixing. So, as always, we can go ahead and start by attaching the various R packages that we will need today to our local environment. Since we have already derived out the signature matrices and we're not creating any new gates, the number of required packages will be fewer compared to other weeks. Namely, we just need to go ahead and load Flow Workspace, Lucernaga, dplyr, and purrr. Once this is done, we just need to go ahead and designate a file path to the location of both our storage and our output folders. So as we've seen in the previous weeks, basically we're providing the full file path location between our working directory and that particular folder. So in this case, if you're following the course's weekly organization scheme, it's going to be week 14, and then within that folder you'll have a subfolder called data and a subfolder called outputs, in which case you're just doing file path the data, saving that to the object storage location, then file path outputs, saving that as the output location. With the storage location of our files now established, we can then identify the CSV and the .fcs files that are located within using the list.files function. So as we've seen previously, list.files, we're passing it our little storage location file path. We're identifying at this point the CSV files, so for pattern we're providing .csv. And then because we want the full file path between our working directory and those individual files, we go ahead and set the full .names argument equal to true. And then at that point, as you can see, we end up with the 3 different CSV files, one for the bead signature matrix, one for cells, and the stashed one also contains some of the unstained. We'll work with these individually in just a little bit. And from there, we just need to go ahead and identify our FCS files. So basically same kind of setup, but we're just changing the pattern to be .fcs. And at that point, we're able to find the FCS files that are present within that same folder. For this example, these full stained files, we just included both the control and the PMA condition for infant 52, which was a cord blood donor, and then control and PMA, which was our adult donor for ND 050. So with that done, let's go ahead and load in the individual CSV files that contain the signature matrices for both the beads and the cells. We can do this with the base R's read.csv function, saving each to their own object/variable. In the process, we can then remove any unnecessary metadata columns that may be still left over from the processing, since in addition to our detector columns, which contain the actual values we need for the signatures, we only need the fluorophore and the antigen information when it comes time for the actual unmixing. So we can go ahead and get that started. Remember, CSV_files is a vector, a tree file paths. The very first one corresponds to the bead signature, so we're just doing the square brackets index 1 to grab it based on the index location. This then, we remember to set check.names equals to false, because we don't want the data frame automatically being corrected in terms of its column names to match in our syntax. We only want to change the column names when we are actually ready to change the column names. And then this gets assigned to the new object that's being created, bead signatures. And from there, if you remember to the walkthroughs for signature matrices and signature comparison, we had a few extra processing columns in terms of name, type, detector, and negative. Those we don't actually need for mixing, so we can go ahead and remove those using the dplyr packages select function, just adding this little negative in front, basically dropping them in this particular circumstance. And then at that point, if we go ahead and check, we can see we're left with just the fluorophore columns that we're interested in for the detectors, and then the fluorophore and antigen metadata columns. And sure enough, if we go ahead and check the actual view of our underlying data, we can see that they're still correctly ordered. We got BUV 395, 496, 563, following that laser in that peak emission wavelength pattern as far as what we saw. And then if we look at the actual values, we can see they have already been normalized / scaled, with everything being between a value of 0 and 1. And then at that point, we can just go ahead and repeat the same process for our cell signature controls, reading in from the second index of that vector and doing all those same steps, just so we can end up retrieving our cell signatures that we derived. And now we have all those main elements that we actually need when it comes time for our unmixing. So before we dive straight in, it's often worthwhile to strategize what are we trying to accomplish first. So over the last couple of weeks, we have derived out the signature matrices for our single color controls, both the beads and the cells. Similarly, we have an additional matrix that contains the unstained signatures. These matrices, in addition to the detector columns, also contain metadata in the form of our fluorophore and our antigen columns. Separately, we have a file path to the location of our individual raw FCS files. Once we load these in individually into R, we need to access their expers matrices slot, where the raw measurement values are being stored. After these end up being extracted, we can then provide these and the equivalent signature matrix columns to our unmixing function, which will carry out the unmixing calculation, and then return to us the respective fluorophore contributions. We consequently would then end up with a new matrix that would contain the calculated abundances of each fluorophore for a given cell. These in turn could be swapped into the existing expers plot within the flow frame object. And then at that point, we could carry out any of the corresponding changes to the parameters in the keyword slots that would then need to be carried out, converting the formatting from one typical of a raw FCS file to that which is expected for a unmixed FCS file. Some of these steps may overlap quite extensively with week 10 bonus material, so we may be able to reuse some of that code instead of needing to write it entirely from scratch. And lastly, we would then need to save our new FCS file to a designated location. At that point, we can go ahead and visualize it just to make sure there was no formatting mistakes on our end when we made the coding steps. And then if none are encountered, we would then be set up for next time when we go in and evaluate our unmixed FCS file to see if we can find any unmixing errors that might affect the downstream analysis process. So, to go ahead and get started, we first need to create a basic function skeleton. In this case, since I'm planning to do just ordinary least squares, I'm just going to call our new function anything that I want. In this case, I'm choosing to call it old-fashioned unmixed. As always, when you're creating a function, individual arguments will go inside the parentheses, and any code that is being run will be run within the curly brackets. So if you see the initial start of this function, you'll see function being assignment arrow to whatever we're going to call the new function, and then arguments will go in between the parentheses, and then curly brackets will be code that's actually going to be run. Currently we have no code, so I just have a little placeholder comment saying what's happening right now. So in terms of what is being passed to our old-fashioned unmixed function's first argument, at the end of the day, we want to simply create the function in a way that we can pass it a vector that contains the file paths to the respective SES files that we want to unmix, and then we just need to stand back and let it handle the entire rest of the process by itself. So we're going to need to be able to iterate, either using the pers, packages, map, or walk functions, or their base R equivalents. Consequently, it's probably easier just if we use x as our first argument. We can then add that into our function by placing the new argument within those parentheses. Additionally, we need to help our future selves when it comes time so that we don't forget what each argument does sometime next month. Actually, who are we kidding? I'm likely to forget by the end of today or by tomorrow for sure. So let's go ahead and write out those actual argument documentations within the Roxygen 2 skeleton, providing the name after the @param tag, which allows us to provide the name of the argument and a short description of what it actually does. At that point, if we are creating a function, we just need to go ahead and run the cell code block if we're working within Positron or RStudio, just to create the function in our local environment, which would then appear in the right secondary sidebar right underneath the session tab. So what does this look like in actual practice? So as you may remember, Roxygen skeletons have the little comment hashtag apostrophe, and there's a little description of what our function is doing, and then the @param designates a argument. You see the name is x, and then we give a short description of what it actually does. And then the changes we've made so far, we've placed x as our first argument, as denoted by being within the parentheses. And then just as an internal check, right now we're just going to have it printed directly to our console every time old-fashioned unmix is run. In this case, since we're passing in a file path, we should see a file path being returned to our little console window. At that point, we can just go ahead and refresh the function by running it again. This upstates it in our local environment, and then at that point, we can set it up with a code block line directly below using the per packages walk function just to iterate through the vector of FCS file paths. So this is what it looks like in practice. As we create our function, we go ahead and run it with our checking output line, and sure enough, we're getting back the file paths that we were passing in through FCS files. So, as we proceed to build out the rest of the function, just remember, anytime you make a change to the function, rerun the code block in which it's found, so it's refreshed in your local environment, so that then you can take advantage of all those changes that you have made to that function in terms of new arguments or alternative things that are being returned by the function itself. So, next up, we're going to go ahead and load in the designated FCS file into our local environment from where it's being stored, being designated by that iterated in file path in terms of that x argument. So if you remember back to week 5, there are two ways that we can load in FCS files into R. We can load them in either as a flow frame object or as a cytoframe object. The main difference between these two versions is whether the object is loaded immediately into our rapid access memory or our RAM or not, whereby it's working via a pointer pointing at wherever it is on our hard drive, solid state drive, NVMe drive, whichever storage device you have. So in the case of unmixing, we need access to those internals pretty much right away. We're not really planning to do any pre-gating, and we're mixing individual objects one at a time. Because of this, it's useful if we just go ahead and read it directly into rapid access memory. So we're just going to go ahead and use the flowcore packages read.fcs function to load in our raw FCS files directly as a flow frame object. Since this means we're going to be relying on another package, this introduces an external dependency. So we need to be able to correctly specify within the function as we build it that it needs to go find what this function does in the respective package. So the way we can do this is twofold. We can add a import from tag within the Roxygen skeleton and then provide the name of the parent package and function. Since we're not yet at the point in the course where we're going to be building description and namespace files, we also need to remember within the actual curly brackets where the code is being evaluated to include the name of the parent function as well as 2 colons so that it's able to correctly locate what's happening in the absence of that package having not yet been loaded to our local environment. So what this is going to look like in practice, as you can see, I've noted the dependency in the @import from. In this case, it's coming from flowcore, and it's the read.fcs function. And then within my code block, within the actual function itself, within these little curly brackets, I'm just making sure that read.fcs is able to find where it's coming from. In this case, designating this function's originally from flowcore, designated by those 2 colons right next to each other. And then at that point, we can just set up the argument for the normal part of the code where we're loading in whatever file is found at the file path X. We're not transforming, we're not truncating the max range, and we're saving this to the raw FCS as the object name within the function, and that in turn is going to be returned to us when the function completes. Sure enough, at that point, if we refresh our code to be able to refresh the function in our environment, and then we run our little testing code block directly below, in this case, just designating the first FCS file path within the FCS_files vector, you see we're getting back a flow frame object being printed to our console window. And then if we look at it right now, we can see it's very much the same kind of formatting that we got it from import. It's infin52's control sample. It has 10,000 cells, 74 columns. All the little individual $P row names designate different detectors that are found in a raw FCS file. And in this case, for the Cytic Aurora, we have 719 keywords being recorded in the description list. Alright, so getting back to our flow frame object, we're getting back to expected flow frame object. So we can then now go ahead and start gathering the actual components that we would need for unmixing. So our next order of business is retrieving the expert slot content containing the raw MFI values. These will be containing both the detector columns as well as some additional columns including time, side scatter, in this case off the violet laser, forward scatter and side scatter blue, which would be off of the blue laser. So to actually go ahead and update our function, we're taking that little intermediate we had, the raw FCS, and we're going to go ahead and pass it to be able to grab the internal expr slot. That would be the expr's helper function from the flow core package, which will access it, grab it, retrieve it, saving us all the hassle of adding all those little @ signs. And then at that point, we're creating a new object called the raw matrix, which we're switching out for the raw FCS, which had been in return, so that instead we're now getting back the raw matrix to our object. At that point, refresh the function, rerun the output line, and as you can see, we're getting back the raw values corresponding to those designated detector columns, as well as time, side scatter, forward scatter, etc. Now, the easiest way of probably being able to select the detector columns from the forward scatter, side scatter columns would be using the dplyr package. So at this point, it's probably just easiest just to convert this matrix object over to a data frame, which we can do, and so that way we are able to use the different tidyverse packages in terms of tidying the data. So let's go ahead and do that. In this case, taking the raw matrix output, we're putting it through the data.frame function, which will convert it to a data frame, and then at that point we can have it returned. And at this point, since we've added in another flow core dependency in the form of exprs, we just go ahead and also update the @import tag from it, adding exprs there as well. At that point, refresh the function, rerun our output line, and we get back a little matrix style object. Obviously, there's 10,000 different rows at this time, so I'm just going to go ahead and use the head function, designating down to tree. And remember, map function returns as a list, which is why I'm doing these little square brackets one, basically grabbing that first value in that list, which is our particular matrix. So with this done, we need to isolate out the detector columns, since these will be the values we'll need for unmixing from the other columns, things like time, side scatter, forward scatter, side scatter blue. We will still want to retain these columns. We don't want to get rid of them entirely, because after we've carried out the unmixing, we want to add them back into our mixed FCS files so we can gate based off a scatter for our lymphocytes, our monocytes, or our granulocytes. Rather than simply specifying each individual column that we want to retain, we can use the combination of the dplyr package's select and matches functions to basically grab a combination of any column that has a column name that matches a particular character strings, or /strings if we're separating them by the little pipe. So in the case of above, we could just basically inside matches place a character string like forward scatter pipe, side scatter pipe, time. And in that case, as it checks the column names, anything that matches or partially matches those character strings will end up being selected for and saved out as its own little data frame. However, for certain instruments or certain cases, there could additionally be other columns that we might want to retain beyond just forward scatter, side scatter, and time. Rather than then needing to go open up the function, modify it directly, it would be better if we had to set as a new argument, in this case a second argument, something called like retain these. And then, because we don't want to have to start rewriting forward scatter, pipe, side scatter, pipe, time each and every time we run our unmixing function, we could just provide a default value of that. That way it's able to do it, and we're just able to handle any edge cases as we encounter them without needing to specify new values each and every time. To do this, let's go ahead and update our argument. We're adding in the second argument, which will be within the little parentheses, and then we'll just need to update the Roxygen 2's documentation for the new argument, as well as tell us what it does. And then at that point, since we'll be adding a couple of dplyr dependencies in the form of select and matches, we need to update them in the Roxygen, as well as internally with the dplyr double colon, just so that they're able to avoid any dependency issues in case the package has not actually been libraried to our local environment when we run the function. So what does this look like? As you can see, new argument being designated by the @param in the Roxygen skeleton, a new @import from, handling those dplyr dependencies, and then because we're building out the code inside the function, I'm starting to add a few comments just to let us know what each of these little chunks of the function are actually trying to do. So in this case, this comment is basically telling us we're trying to identify the detector columns, which will make it much easier when you're trying to reread what the function's doing later on in the future. So at that point in time, we can see we're taking our raw data frame, we're passing it to the dplyr select function, which is using matches to match against the retain any pattern that's seen in retained Ds in the column's names. And because retained Ds is equal to forward scatter pipe, side scatter pipe time, any of those columns will end up being selected and isolated out into the new object stashed columns. And then at that point, as an internal check, I'm just taking the column names, creating them as their own object, and then having return give us back that object so we get a vector of what has been stored in stashed columns. So at that point, refresh our function, rerun our code block, and as you can see, it is correctly partially matching based on the values we provided. It's retrieving time, all the different side scatter variants, and all the different forward scatter variants. Alright, we now have a generalized way to designate which columns need to be kept outside of the unmixing process. And at the same time, if we take that existing line of code that we're using to select these that are being excluded, if we add a little exclamation point inside, we can basically flip the argument and end up grabbing all the columns that we actually want to keep, i.e. all those detector columns. So, what's that going to look like in practice? Basically, right where we had stashed columns, we just copy this line here, go ahead at that point, paste it, change the name to working columns, and we're just adding in that little exclamation sign to flip the order. So if it matches forward scatter, side scatter, and time in that case is not going to be kept. If it doesn't match them, it's going to be kept and it's going to be saved to working columns. And as you can see at this point, we're successfully isolating out our individual detector columns that we're going to need for the unmixing process. So, in the case of the Cytek Aurora FCS files that we have for this example, this very last line of code would have been sufficient to isolate out the detector columns and we could proceed downstream to unmixing. However, even on Cytek Auroras, there's an instrument setting that would have allowed us to also acquire height and width in terms of our detector parameters, and this is default on other manufacturer instruments as well. So, we may need an additional step, basically excluding out whichever variant of area, height, and width we don't want to be using as the signatures. So in this case, if we want to be using our signatures based on the area parameter, we would want to be excluding the hyphen height or hyphen H, the hyphen width or the hyphen W, but keep anything that ends in hyphen area, i.e. hyphen A. So, once again, easiest way to do this will once again be a new argument, but also setting a default so that we don't need to actually worry about it on our everyday basis. So we're going to go ahead and call that third argument detector exclude, and in this case, since we want to kind of just go ahead and keep those hyphen A column names, we're just going to have the default value be hyphen H and hyphen W to handle any of those edge cases as they may be encountered. Additionally, at this point, we just need to go ahead and update. So as we scroll downwards, we can see as we go down here, we're setting up a new working columns. And from the new working columns, we're just setting this one to exclude for detector exclude columns across the board. So at this point, in this case, we're not really isolating out anything separately. We already have the hyphen A selected for. So in reality, we're just actually keeping what we already have present and moving the rest out. And at that point, just refresh the function and just go ahead and proceed. And in our particular case, we already have the same column selected. We didn't need to make any changes, so it was all set to go. But if we had one of those edge cases, you could see it's still retaining the columns that we're actually interested in. So, having preempted a potential issue and still retaining the detector columns of interest, let's go ahead and swap out the return content to be a data frame instead of the column names checks that we've been doing so far, so that we can actually see the underlying experts values that are being retained. So at this point, everything else remains the same, and then in this case, we're just switching it out. Instead of returning the column names, we're now returning our working columns object within the function, and then at that point, we should be able to see the experts values. So at that point, refresh the function, rerun our output line, and we're back to seeing just the detector columns, the things that we actually need in time of our unmixing. Alright, so from our raw FCS file, we have retrieved out the expert values that we'll be needing for unmixing. So within this data frame, each row basically corresponds to the detector measurements recorded for a individual cell. During unmixing, each row is unmixed versus the reference signature matrix, which allows us to estimate the presence or the absence of each fluorophore, as well as its relative abundance. When unmixing is then complete, we end up having a matrix object with the same number of cells, but instead of the detector columns, we have new columns that will correspond to the fluorophores that were provided within our signature matrix. This matrix object is then ultimately added back into the expert slot. So beyond providing the signature values that will distinguish each fluorophore from each other, the signature matrix that we end up providing also contains right now the source of the fluorophore and the antigen names. So first off, we need to go ahead and provide the signature matrix to our old-fashioned unmixed function, which we will need to do via a new argument. In this case, we're going to go ahead and call that one signature data. And then also we want to retain some flexibility in terms of whether we provide this directly as a data frame object or just a file path to a CSV file somewhere in our system. So we could set this up with a little conditional statement, the if, else, or else if kind of statements, depending, so it would handle whatever type of object we end up passing to signature data. So what this is going to look like in practice, we're adding a new argument, so we're documenting it within our at params. And then at that point, placing that new argument between the parentheses. And as you can see, we're starting on a new third little chunk of code inside here. So I'm just going to give it a new comment saying what we're doing. And then at this point, we're starting to write out the conditional. Remember, a conditional is only evaluated if whatever's between the first parentheses here comes back as being true. So if the signature data object we pass in is a data frame, this will come back as true. And then at this point, the code within the little curly brackets will be evaluated, in which case signature data is just going to get saved to signatures. Alternatively, if the object we passed in is not a data frame, let's say it's a file path to the CSV file, it would then pass it to read.csv. It would then pass off to the else statement, which will just go ahead and immediately read in. So anything within those curly brackets will now be actually executed. So as you can see, read.csv would read in the file path, check.names is set to false, so we're not changing any of the column names. And then this is all then being saved to a little signature object, and that's what will be used by the actual function itself. So note, regardless of which path is actually happening, we're still ending up with the same kind of name, signatures, so that way there's no deviating forks in what's happening inside our function. So at that point, if we go ahead and run, as you can see, we're reading it incorrectly in the context of if we read in bead signatures, and we're all set to go at that point in time. Alright, with this accomplished, we still need to separate out those character columns that will contain the metadata for fluorophore and antigen from the numeric columns, i.e. the detector columns that are being used for the unmixing. So we can simply separate out the character columns used for labeling from the numeric columns which are being used for unmixing. We can do this through that combination of select, where, and is numeric that we have encountered previously throughout the course. So what this is going to look like, well, we're adding in some additional new dplyr dependencies, so we need to update those in the Roxygen skeleton, and same thing, the base package utils, read.csv that we had a little bit earlier. And then down here, right after we go to our conditional statement, we're basically separating out our metadata from our numeric columns. In this case, piping signatures true to the select function, which is using where and is numeric to identify things that are numeric. If it's a numeric column, it will evaluate as true, but that little exclamation point will flip it to false, and it won't be retained. So only character/logical columns will end up being stored within the metadata. And then vice versa, if we don't include that little exclamation point, it will, if it finds a numeric column, it will end up keeping it, and it will end up storing it within numerics at that point. So if we go ahead and check, we can see that within the numeric columns that are being stored, we still end up with just the signature values as far as the matrix goes for the different detector columns themselves. So our signature retrieval functions that we've been working with over the last few weeks have the ability to return either the raw values or the normalized scaled values that will range from 0 to 1. For unmixing, we're going to need these scaled values. So we can add an internal check through the use of an additional conditional statement that if it gets triggered by the presence of a value that's greater than 1, we'll just proceed to normalize the rest of the values in the matrix before proceeding. And let me double check before proceeding. Yep. So what this is going to look like in actual practice, as you can see, is if any of the values within numerics is greater than 1, At that point 1, it's going to evaluate as true, and it's going to proceed to within these little curly brackets. And then first it's going to message you to let you know, hey, it found values that were not yet normalized. It's going to start normalizing them, just in case this was an error. And then it's just copying and pasting in the code that had been used for a spectral signatures lesson as far as the actual scaling normalization process. Obviously, if the values have already been scaled, it doesn't need to rerun this line of code. So at that point, it would just not actually be evaluated. And then at the end, we're just overwriting the intermediate normalized values, putting it back into the numerics name. So that way, both of these coincide back to where they need to be with a numerics value data frame that has values between 0 and 1. So, refresh our function, rerun our line, and as you can see, nothing has changed in our particular case because all these values had already been normalized. And before we dive into unmixing, we should go ahead and make sure that the detector columns that are present within our experts object end up matching the total numeric columns that are present within our signature matrix, so that we avoid having our code fail out without any really understandable explanation about why. So the simplest implementation of this would be to add one more conditional statement, basically using all and then column names as shown in the next check. So as we scroll all the way down, you can see we're adding in a new comment tag telling us what we're doing here. We're making sure the dimensions match each other. So basically, if the column names of the numerics data frame are equal to the column names of our working columns data frame, and all of them are just matching each other, we flip them. So they're not matching each other in that conditional statement. We just go to immediate hard stop, and we're going to get this little error printed to our console window. So at that point, we need to go ahead and double check to see if this works. Rather than try to bring in a mismatched file just to do this test, we're just going to kind of use base R, take our existing bead signatures data frame, and then just drop the very last column. As you can see, this is basically grabbing from the first to the last column, but -1. So it's just going to grab the first 28 or so 404 columns. I'm going to pass that in, and then at that point, we're going to have a mismatch. So if we actually run the code, you'll see we get back an error that looks something like this at that point in time. So then at that way, rather than just a little silent error that doesn't explain what's going on, you know, oh, I'm still having extra numeric columns that shouldn't be there, or I'm trying to unmix this with something that was acquired on a different instrument, so at least you have some idea of what's going on before you move too far into the process. So with this check now implemented and our starting components collected, we are ready to actually proceed with the actual unmixing. And let me take a sip of coffee. Alright, so when we are mixing, we need to go from the total signature measurements from the individual cells, and referencing the signature reference matrix, calculate out the abundance for each fluorophore that best fits the signal that's seen on the cells. So this process is often mediated by various least square methods, the more frequently encountered of them for spectral flow cytometry data being ordinary least squares. In R, this is implemented in the lsfit function from base R stat package. So to carry out this task, both the signature values and the sample values need to be in a long style format, being more rows than columns. Since both of our data frames currently are in the wide format, having more columns than rows, we need to swing/pivot them to be in the correct orientation. So to avoid issues, let's go ahead and back up the column names that correspond to each detector, just as a precaution, but then at that point we can go ahead and transpose each matrix into the long style format, using the base R's transpose, i.e. T parentheses function. So at that point, as we scroll down, we see new section that's going to be down here, and we're just saying go ahead and save those column names just so we have them in case for later, and then just transposing the numerics, transposing the working columns, saving them as their own variant objects so that we're able to work with them later. And at that point, we can just go ahead and internally check, and sure enough, instead of having our detectors on the columns, we now have our detectors showing up in the row names, and we ended up having to just select a little subset of the columns because we had 10,000 now in terms of what we end up seeing. So this is why we have this little complicated visualization code line here just for the website's sake, so it's not 200 or so pages of just output lines across the board. So with both our signature and sample values now properly oriented, we can actually run lsfit to carry out the unmixing process. So what this is going to look like, as I scroll all the way down, you can see I'm actually doing a new comment telling us this is where unmixing is actually starting. So in this case, for least squares, we're running lsfit. And in this case, after all that build-up, all this code to get here, the actual unmixing itself is rather simple. X is being passed transposed signature values, Y is being transposed the transposed sample values, and then we're using an intercept is equal false argument. And in this case, this will go ahead and run and carry out the unmixing, and it will return to us the list that we actually need to parse in to get the actual unmixing values that we need. So we get back a named list. If you may remember, we can use the names function to basically see what the individual items of the list are. So we can go ahead and do that. So in this case, for that very first one, we can see we get back coefficients, residuals, intercept, and QR as different parts of those named arguments in that list. So let's check these sequentially. So if we check within lsfit's named list return, the very first entry is the coefficients. So if we run dimensions, so the dim function, we get back the following. So we end up having 28 rows and 10,000 columns. This 10,000 is going to be the number of downsampled cells that we started with. 28 is kind of similar to the number of fluorophores we had, but it's not quite what I'm expecting. This particular panel was 29 fluorophore panel, but what you may remember is our signature matrix that we've been using so far has been the bead signature matrix, which didn't include the zombie IR control, which wasn't present at that point. So if we actually were trying to unmix with the full panel, we need to actually go back and add in zombie IR to our bead signature matrix, because as we currently ran it, it's not actually accounting for all the signals that were present in the cell. We can also quickly go ahead and check to see the first few rows with the head function, just to make sure everything looks normal and that our matrix is not full of NA or null values respectively. So at that point, if we go ahead and run it, yep, we got numeric values coming back, so this looks as expected. Next up, we have residuals. Let's go ahead and check them with the dim function as well. In this case, we see there's 64 rows, 10,000 columns, so this is matching what we had for our raw spectral data before we actually unmixed. And we'll circle back and see how to actually investigate these residuals, because there's some data that can be visualized from them that might be useful to know about, but we'll check on this a little bit later on. The intercept was one of our arguments we had specified within the line of code that we ran. As you can see, it's set to false. Similarly, if we check what's actually present in that named list output, you can see it's also showing up as false. So it's just being stored as a reference. And then same kind of story for QR. QR is also a list, so if you check the names of the items within QR, you can see we got QT, QR, and a few others. These are more on the minutia side as far as what this function does, so we're not actually going to walk through these today, but they're there for anybody who would be interested in them. Alright, so overall, it's the coefficient values that we will need to be grabbed because they correspond to those unmixed fluorophore columns that we want to be able to grab and place into our new unmixed FCS file. However, there's some additional useful information within the residuals that are worth taking a look at, since some of the more newer, complicated unmixing methods end up taking advantage of these values when it comes time for their own calculations. So first off, let's go ahead and write a small helper function just to help us visualize the residuals. We're going to go ahead and call this one residual plots. Basically, it can just take directly the lsfit output, which is going to be that list, and then it can work from there. So its only argument at that point needs to be least squares fit. So at this point, as we write out the Roxygen skeleton, we can see these are the elements. Parameter, as far as argument, is going to be least squares list, and then saying what it actually does. And 1 second. And then at that point, within the actual function itself, we're just going to go ahead and do a lot of ggplot code. So in this case, I'm just by convenience going to call library on ggplot2. We're just passing in a named list, so we're just actually grabbing the residual component, and then we're deriving out some individual bits and pieces, like which are the detectors for observations, and then the root mean square, which is going to be the fit for the residuals, and then those are just being passed directly to the actual function itself. And then at that point, if we run the function in our local environment, we now have residual plots available to be used. So we can just go ahead and add that into old-fashioned unmix as the next line after we do the unmixing. So from our least squares named object that we get back here, just passing it indirectly, we're getting back our little plot, and then we're setting the plot to be returned to our local environment. And then at that point, we can just go ahead and plot residuals for all these FCS files so that we're able to see the comparison. So if we go ahead and run it, you can kind of see we're getting back a residual style plot running from 0 to 64, which corresponds to the number of detectors that we had. And in this particular case, you may remember we forgot to include zombie in the IR, so our residuals are kind of high towards the last few detectors, which would correspond to where we expect to see the peak fluorescent emission for the zombie in the IR fluorophore. So that's pretty cool to see, and it's one of those reasons why looking at the residuals can be interesting. So let's take a closer look at that in the next couple examples. So as we saw, we had more residual leftovers when it comes to the later half of our detectors where we expect to see zombie in the IR. Let's actually see about adding in the zombie in the IR signature as well as our unstained signature for autofluorescence in alongside those bead signature controls, bead signature references, so that way we're able to account for all the signal that is actually present on the cells. So how are we going to do that? Well, we're going to go back to that third CSV file in the vector that we haven't loaded in, go ahead and read it in as alternate cell signatures, process it down to get rid of those intermediate columns, and in this case we want to grab just our unstained, so basically using the combination of the dplyr filter and stringer structure detect functions. For the column 404, we can identify what row is named as PBMC unstained, isolate it out, and save it to the unstained data frame. And then we can repeat that same process at that point for zombie in the IR, isolating out its respective row, and then saving it out to the zombie data frame. So at that point, let's go ahead and start by just adding back in the zombie signature to the bead signature matrix, and then compare the before and after residual plot to see what happens when we actually remember to include all the signatures. So to add it into the existing signatures, we can either use our bind or the dplyrs bind rows function, just combine them in together. And then at that point, updated self-references is then going to be passed to the signature data argument. We rerun this at that point in time. And then at that point, if we remember our before, and this is a little plotly interactive plot, so give me a second as I zoom out. If we remember before we remembered to include zombie in the IR, we had residuals that were pretty high on detector 62, around 8,000 for the RMS. But if instead we had remembered to include zombie in the IR, if I go out and zoom out, for that 62 detector, you can see it's substantially reduced in terms of its residual. It's down to 300 something. So all that massive amount of signal is now being accounted for correctly. And the leftovers are more in a couple other areas as far as whatever peak detector 50, 39, and 6 are respectively. Alright, so we no longer have that peak around where we would have seen it before. So what happens if we also remember to provide a autofluorescence signature to our signature matrix? So we just need to go ahead, instead of just adding back in zombie to the data frame, we can just go ahead and bind rows to include the unstained as well. At that point, we make sure this new updated data frame is being passed to signature data, and we can go ahead and rerun. And if you remember, when we did include zombie, we were just having some of those leftovers corresponding to the 6th detector at 800, 39 at 1176, 50 at 1106. But when we remember to include both unstained and zombie, if you look at our residual plots at that point, we still have this one over here at 50, but then 6 is substantially reduced, as well as the 39 one's a little bit down. So we're not actually quite sure where this signal is coming from for this respective specimen. There's probably something else in our system we're not accounting for correctly, but we're actually reducing the total amount of residuals that are actually present. So we have had some shifts across the board, but we've improved substantially compared to that original condition where we had forgotten to include these two signatures in our matrix. Now, this was a use case where we had forgotten something unintentionally, but what happens if we intentionally forget to include something? And let's say that was something that was very important for our panel. So in this case, if you remember, in this particular panel, we have BUV805, which is going to be CD4. This is a mainly T-cell focused panel, so it's going to be a lot of actual signal coming from this fluorophore. So if you are looking at the existing data frame, but you use little square brackets, in base R notation, if you do -7, comma, it's going to remove that 7th row, which in this case would have been BUV805, which is basically going to subset down our data frame to not include BUV805. If we end up passing that one and carrying out a mixing and then looking at the residual plots, what you're going to see at that point is when we forget to include BUV805, just like we saw with zombie NIR, the corresponding increases where we expect to see them end up appearing. And as you can see, although the residuals are not the main output that we need from LSFit, they can be quite useful in determining whether all the signal was properly accounted for within our signature matrix or not, which is why some of those more complicated unmixing methods end up taking advantage of it. So, before we turn back to coefficients, i.e. those unmixed outputs that were our main reason for being here, let's add one more argument that would allow us to get back to residual plots if we wanted to use them in the future. We can call this argument return type, setting the default to FCS, but also remembering to include a residual condition option to return these plots to us if we so wanted to see them and visualize them later on in the future. So, once again, new argument, new update into the Roxygen 2 skeleton, and we place it as well as its default present within old-fashioned unmix. And then at that point, as we scroll down where we had been returning the residual plots just as an individual line, we're now wrapping those within a conditional statement, basically saying if the return type is equal to residuals, it will go in, run the residuals to get them as a plot, and then it will just short circuit the function, returning the plot directly to us in our working environment. And if the return type is not set to residuals, if it's set to FCS, it's not going to evaluate this conditional, and it's going to continue down with the unmixing process to give us back eventually our FCS file. Sure enough, at that point, we refresh our function so it's active in our local environment with the correct new arguments. And then at that point, if we try the return type is equal to residuals and run it, you see we get back our little residual plot. Vice versa, if we run it with .fcs as a return type argument, we get back the values that we expect for our unmixed FCS file. Alright, so our main reason for being here, the coefficients, i.e. the unmixed data, let's go ahead and turn back to them. So you remember we had to pivot from a wide format to a long format to be able to carry out the unmixing. If we want to be able to put them back into the expert slot, we're going to need to pivot back to a wide style format, so we're going to need to retranspose them all again. So at that point, as we scroll downwards, we're now back into trying to fit everything into the correct format, so we're just going to call this little comment line reassembly. We're just going to go ahead and transpose the coefficients, bringing them back into the expected kind of format. And at that point, before we move too far, the last argument we used was using the bead signatures data. We do remember we have both zombie NIR and unstained signatures that we're trying to add in to be able to unmix correctly, so we need to include these in the signature data. But remember, based on the individual number of rows that are there and the particular order of those rows is how our actual final FCS file is going to be visualized. So if we don't want zombie NIR tacked down at the very bottom after APC Fire 810, if we want it in the correct location based on laser and peak emission, we're going to need to rearrange this data frame before we move farther. So let's do a little bit of data tidying just to make sure it's cleaned up. So once again, let's just check. If we take our existing data frame and pull the fluorophores, you can see our current problem. Zombie NIR is in location 29. It should probably be right up here between APC R700 and APC Fire 750. And then PBMC unstained is quite long for just an autofluorescence designator. So we can just rename that to something like autofluorescence, which would also do the trick. So, to actually get the rearrangement of the rows, we just need to write a small helper function to do so. This is the example of what it actually does. But in practice, basically, it takes your data frame, it takes whatever's located at row 29, and then it will relocate it after whichever row you're designating. So in that case, it will move zombie NIR to the correct location. At that point, we're passing it into the dplyer's combination of the mutate and case when, so that when the 404 column defines a row that is called PBMC unstained, it's just going to rename that one as autofluorescence, and then keep any other values that don't match PBMC unstained as they currently exist, which is what the true tilde sign fluorophore is doing here. So then at that point, since these are over on the red detectors, which is at the end of the data frame, we're just running tail for the last 5, and as you can see, we have APC R700, APC Fire 750, Zombie NIR in the correct location, APC Fire 810, and then finally autofluorescence. So at that point, refresh our function, rerun it, and then we're still getting back as expected, so we're good to go in that regard. So now that we're back in our wide format, let's go ahead and we're back in the wide format that is expected for XPers. However, our particular data frame we're working with right now doesn't have any column names, so it's time to go ahead and take that vector of fluorophore names and paste them back in. One thing to note for Cytek Aurora instruments, when you're carrying out unmixing in SpectraFlow software, the fluorophore name ends up having a designated letter at the end, in this case often like -A, designating which parameter was used in the context of the unmixing. So you would see something like FTSE A, Percy P Sci 5.5A, etc., etc. So we should probably do the same just so we don't encounter any weird, quirky bugs if we ever try to bring these files into the commercial software later on in the future. So we can go ahead and try to generalize this process so we don't have to create yet another argument that we have to remember exactly what it does. We can kind of generalize this based on what column names were actually being retained at the time we went to the unmixing steps. So what this is going to look like, no new arguments, we're just scrolling all the way down. And then at this point for reassembly, basically we need the fluorophore names, so we're going to retrieve them from the metadata. We need those working column names that are going to be the detector columns, so we save those to the detector column names. Using the sub function, we're basically getting rid of anything in the first part of the individual names for the detector columns before the little hyphen mark. That way we're keeping just the last ending of it, which will be -A, -width, -height, whatever the case may be. And we're just keeping the unique value, which will be the letter we want to append on. At that point, we can just take our vector of fluorophore names, paste on with paste 0 the actual appended letter, so -A or whatever it may be. At that point, we have the corrected 4 fluorophore names, which we can then append, use to replace the existing column names and transpose least squares. At that point, refresh our function, rerun, and as you can see, we've updated the column names accordingly based on what we're expecting to see. So, with our unmixed fluorophore columns now set, we can take those stashed columns from very early today, time, side scatter, forward scatter, side scatter B, and then just go ahead and add those in alongside our existing fluorophore columns. So as what you may remember, down to the bottom, we can do this with either cbind from base R or bind columns from dplyr, but basically we want time to be the first column just because that's where it was in the raw FCS file, so that will be stashed columns as the first entry, and then adding the fluorophore columns after we've added in this data frame here. And this will be our unmixed data object, which is what we'll end up returning to our local environment. And at this point, as you can see, time, side scatter, forward scatter, side scatter blues, and then the actual fluorophore columns appear in sequence. And with that, congratulations, we have now unmixed our data into a format that can then be returned to the XPers slot. So at this point, we have assembled a data frame that contains the unmixed data for each cell in our sample. However, if you remember the week 3 and week 10 walkthroughs, you may remember how interconnected the various elements between the flow frame slots are, namely XPers, parameters, and keyword. In this case, since we are instead of creating a new FCS file entirely from scratch, we need to retain certain aspects of the original raw FCS file metadata. However, we also need to remove all the metadata that's associated with the new detector columns, which are no longer present, and add any new metadata for our newly unmixed fluorophore columns so that they are correctly visualized when it comes to term for commercial software. Since there are multiple moving pieces, this is clearly an area where you need nested internal functions to be able to carry out the job. So for anyone who nerds out about FCS file internals and wants to see the additional details, there's a bonus walkthrough. We recorded that one on Tuesday as well, so it's in the video format as well. But for the vast majority of you who'd rather not dive into all the process of converting a raw FCS file's metadata to be the correct unmixed FCS file's metadata, the code is just included in the code chunk below, as well as that function is included as a .r file within this week's R folder so that we're able to use it again in the future. So just to terrify or intrigue you, depending on which side of that argument you end up on, this is unmixed internal. So as you can scroll, there's a little complicated things as far as cleaning up the description list, making sure the parameters are updated, and then finally pasting them all in together into a new flow frame object. And of course, it also has its own little helper function to mediate the process. But in case of if you don't want to deal with these internals, you just have to run the code chunk that they're located in that makes these functions available in your local environment, and therefore you're then able to use them within old-fashioned unmix. So let's go ahead and do that by adding in unmixed internal, which is returning us the properly formatted flow frame, so that then we can just proceed downstream for it. So at that point, if we scroll all the way down here, you can kind of see we left off with the unmixed data with both time forward scatters and the floor columns. At that point, we passed that unmixed data along with the original flow frame and the panel data information to unmixed internal. It's returning to us that properly formatted flow frame, which is being stored under new_FCS, and then new_FCS is what's being returned to our local environment. And at that point, if we go ahead and run, this is what we end up with. So if you may remember, we still have the same name of the original file. In this case, we have 10,000 cells, but instead of that 60, 70 something columns, we now have 40. And then if you look at the individual row names, we now have down to P40, but we can also see the floors and their respective antigen are integrated in. And instead of those 700 keywords that were present for the raw spectral flow cytometry file, we're now down to 443 keywords total. And with that, we are in the final stretch. So, with unmixed internal now taking our unmixed floor data frame and returning to us a properly formatted unmixed flow frame, all we need to do is any of those last minute formatting steps in terms of file name keywords, as well as designating where we want the FCS file to be saved on our computers. So, for renaming, let's give it an option so that we can rename on either one keyword or two keywords, like if we wanted to use both group name and tube name in the final file name itself. Additionally, we want to be able to avoid accidentally overwriting the raw FCS file, so we should add another argument in terms of add-on, which will append on the _unmixed before the .fcs in the name so that it's all working properly. So, what this is going to look like, we have two new arguments, so sample.name, which has some defaults of group name and tube name. So if you're not on a Cytek Aurora and you don't have these keywords, you would need to change the argument respectively, but it's on the argument side, so it's easier to do. And then in terms of the add-on argument, the default is going to be unmixed, and it just tells you it's going to place it right before the .fcs. So at that point, you can see defaults being specified accordingly for those two arguments. And as we scroll down to the bottom, right after we've unmixed, right after we got back the properly formatted flow frame, at this point, we can set up the conditional depending on if the argument we're passing to sample name corresponds to just one keyword or if it corresponds to two. So let's say we're only wanting to rename the file based on group name. That's just going to be sample name is going to be a length of 1, so this is going to be evaluated as false. It's going to jump down to the else, at which point keyword is run on the flow frame, and it grabs for that particular keyword name, and that's what the name is going to be later on in the future. Vice versa, if our keyword, we provided two keywords that we want to use in the final unmixed file FCS name, it breaks that process into separate components and then just pastes them together, putting a little separator of the underscore at that point. Ultimately, regardless of which path is taken, you'll end up with a new name. This name is then appended on with the add-on so that way it's present as far as like the unmixed component. And then once this has been carried out, it will go ahead and add on the .fcs portion of the file name, and then it will just go ahead and overwrite within the keyword list the GUID and the fill keywords respectively to match the new name that we're giving our new unmixed FCS file, so it's not wandering around with the old raw FCS file's name. So at that point, if we refresh our function, rerun it, as you can see, we're no longer with the original name. We're now showing up with a new name that also is denoting that it's an unmixed FCS file. And last, but definitely not least, we need to designate where to store the file. We can copy in some of the previous code that we used as part of the week 10 to simplify the process. What this is going to look like is if we copy in, paste that code, we need to add in one new argument, in this case outPath. And as you may remember, outPath is set as a default of null, which basically means if the situation where you have a designated location you want to store it to, you give it a file path, outPath is no longer going to be null, i.e. it's no longer going to be empty, so it will actually carry out that process. However, in a situation where it's left with the default, if the outPath is null, which is going to be evaluated as true, it's just going to go ahead and evaluate this line of code. It's going to go grab what our working directory location is, and it's going to assign that out to outPath. And then at that point, it's going to combine with our existing name into a file path argument to build out where it's being saved to. And then at that point, depending on what our return type was, if our return type is equal to FCS, it will go ahead and write it out as a new FCS file, saving it to that designated location. Otherwise, it's just going to return to us our little throw frame object to our working directory. So, refresh our function, go ahead and rerun our output line, and this is where our moment of truth happens. We need to go check to see if it's actually been created within our local directory, and sure enough, our new FCS file is there. So we have successfully created the FCS file. However, we still have one more moment of truth. We don't know if we have created a unmixed file that works successfully. So at that point, probably the best way of doing this quickly and reliably would be if you have commercial software, open up your commercial software. If not, go to floriada.io or online, open it up just to make sure, are all the parameters showing correctly? Is everything visualized in a normal way, or is it all a teeny tiny dot way off in the distance? All those kind of quirks that might potentially happen. So at that point, as I load it into floriada.io, I'm looking at the little drop-down menu. All my markers and antigens look to be correct, so that's one check in our favor. As I go and look at forward scatter by side scatter, they are still there, so that looks good. And then at that point, if I look at the individual floors on the two-dimensional axis and put on a logical or a bi-exponential transformation, this looks reasonable enough considering we haven't actually checked the signature matrix. So to all outward appearances, it looks like our FCS file is properly unmixed as far as the formatting goes. So no immediate red flags. We have been successful in our quest of basically taking raw FCS files that we retrieved from import and completely unmixing them entirely within R from start to finish. So as was mentioned earlier, our signature matrix that we use for unmixing has not been fully optimized as far as whether the signatures were the best candidates. So that's something we need to evaluate how our unmixing looks, and based on any encountered unmixing errors, update our decision making as far as which signatures we want to include in the unmixing matrix. And that will be something we'll start touching on next week. For the context of this primary walkthrough, for now, the main tool in our unmixing arsenal is now set up to handle whatever ultimately optimized signature matrix we decide to move forward with in terms of unmixing these files. Additionally, beyond using ordinary least squares, there are other methods to unmix. Some, like non-negative least squares, are actually rather simple to implement. We would just need to swap out lsfit for the actual base R function and just paste it in directly. Others are a little bit more involved in terms of the outputs and inputs that they're expecting, some of which was covered, for example, in the auto spectral and the TrueOS community walkthroughs. Alright, takeaway time. In this walkthrough, we have culminated some of the elements we have been building out over the last several sessions and actually unmixed our FCS files. This is not something everyone can say they have done, so congratulations on having made it this far. As you saw from the process, the main elements involved are actually the pre-processing the data that we need to get unmixed, and then after we actually carry out the unmix function, which is rather simple, reformatting it into the format that is expected for the new unmixed FCS file when it comes to those new columns. So the actual unmixing function at the very heart is rather swappable, which is why you're often able to simply swap out the step which is rather slow running in R for faster implementations in lower level programming languages like Fortran, C++, or Rust, which is what several of the R packages that implement unmixing are actually doing behind the scenes. So next time in the primary course material, we'll be looking at how to evaluate our mixing outputs, and then the following week after that, we're set to look at various algorithms that can help us screen these unmixed FCS files for instrumental issues that might have arisen from fluidics or laser instability. We will also explore how some of these additional comparison metrics in the forms of spreading matrices, as well as the standing index within bonus content that is set to go out over the next couple weeks. Until that time, cheers, we're now at the halfway mark for the original conception of the course. In terms of additional resources, a lot of them will be Peter Mage related. Of the different talks, I know the Flowtext one last year was pretty good, but then the one I particularly enjoyed was the Mathematics of Spectral Unmixing, which was given at the Baberheim Institute Spectral Symposium back in 2022. There was a shorter version of this for the Cytometry walkthrough, so I'm providing that one. Several years ago, David Lecaire at Chicago, as well as David Novo, had that Chug podcast about spectral unmixing. I'm referencing that one as well. And then for anybody who's more interested in the mathematical side, I've referenced both of the David Novo papers that were useful to me when I was first learning about the process. Take-home problems for this week, there are 3 of them. So within old-fashioned unmix, go ahead and modify the return type argument so that instead of returning a .fcs file or a residual plot, that you can also end up getting back a data frame or a flow frame, since both of these are useful potentially if you're trying to work entirely within R and not actually trying to export out. Additionally, feel free to add whatever conditional statements or arguments that you deem necessary to make this actually more feasible on a day-to-day basis. Week 2 is a little more involved. So basically today we looked at residual plots, and we saw that case where we left out BUV 805, and we saw a corresponding fairly large increase on the detectors that corresponded to that fluorophore within our residual plot. So go ahead and rerun the residual plot commands for whatever fluorophore in our panel you decide you want to leave out, just so you can see what happens for the residuals when you leave out this fluorophore. Then rerun the residual plot, but for the full signature matrix to see what it should have looked like. And then instead of doing residual plots back, do the same process for the signature matrix without your fluorophore of interest, but get back the FCS file, and then do the same process with the full signature matrix, getting back that FCS file. The main focus of this question is, we see this increase in residuals for this particular plot, but what is the actual effect of leaving out that unmixed file in the that signature in the unmixed file? Like, is the extra bit of residual not actually impacting how you perceive the unmixing? Is it causing just a lot of issues downstream that are immediately obvious? Is it somewhere in between? It's kind of like a little bit of a treasure hunt to try to figure out how to contextualize what these residual plots actually mean in context for your data itself. And then option 3 is very much a treasure hunt. So one of the first packages that I first encountered that implemented unmixing in R was Christopher Hall's Flow Unmix package. There has been several additional R packages that have also implemented their own ways to unmix using various methods. All these different R packages vary quite widely of how they process their unmixing controls to ultimately get to those signature matrices, but at their heart, the basic unmixing steps are still essentially the same. So at that point, if you were just trying to change your unmixing method from LSFit to some of these other methods, it's just oftentimes just a matter of figuring out what the correct function is and just pasting it into your own function at that point. So at this point, let's go ahead and take advantage of the open source nature of these projects, and let's go on a treasure hunt. So for these 2 packages, both FlowMix and AutoSpectral, go ahead and dive into their GitHub repositories, open up their R folder. Generally, the unmixed functions are saved under a .r file that's called something along the lines of unmix something or other. So locate these unmixed.r files, go ahead and open them. Try to practice your way of reading through the code to see if you can understand what's going on until you find the function that you suspect is actually carrying out that unmixing process. At that point, look up the function, see what's the original package it came from, check its help file, and then take a screenshot and report back as far as what you see. and then feel free to rinse and repeat for however many unmixing methods that you may be interested in, regardless if it's ordinary least squares, non-negative least squares, whole weighted least squares, whatever version of them that you're interested in doing. And with that, those are the take-home problems for today. I'll be in the chat for a little bit, and if you have anything that comes up later on in the week, please head to the discussion forum, and if not, I will see you next week.